here I'm going to review the process of translation, which is the process that makes a protein from an mRNA template. So if we take a snapshot of what generally is going on in translation, again, first we're starting off with an mRNA template that is going to be read 5' to 3'. A ribosome will have been positioned onto the 5' end of the mRNA during initiation, which we'll look at in a little bit. But again, if we're looking at a snapshot while the process is happening, the way that it should look is that there are 3 specific positions within the ribosome that these special RNA molecules called tRNAs can bind. Really, what tRNAs are binding to are the codon sequences within the mRNA. So each one of these is going to be a specific codon which corresponds to a specific tRNA, which has a specific amino acid. So codons are found in the mRNA, and the tRNA has anticodons, which are going to be complementary to the codon. That allows for the code, the nucleic acid code, to be translated into amino acid code. So tRNAs can be charged or uncharged. Charged tRNAs have a specific amino acid attached to them, and uncharged have lost their or donated their amino acid to the growing protein chain. So during the process of translation, this growing protein peptide chain is going to be transferred to the incoming amino acid here. And that allows for the chain to continue growing at this end of the protein. Okay, so before we look at the specific steps of translation, I just want to make sure that we're thinking about where this is occurring. So remember, if we were in an E. coli cell, the RNA would be made, let's say these are mRNAs. They could be floating out in the cytoplasm, or they could be actively being transcribed like this. And there will be ribosomes that can hop onto those mRNAs and start translating them, either away from the DNA out in the cytosol or at the ends of the just being transcribed RNAs. And again, those should be making their little protein chains, once they start doing translation. So these processes are coupled. They can all occur at the same time, or they could occur separately. It just depends. This is distinct from what you see in eukaryotic cells, where the processes are uncoupled. So remember, the mRNAs are being made in the nucleus where they need to be processed in order to actually leave and enter the cytosol. Okay, so here, these mRNAs are going to either encounter free ribosomes, cytosolic ribosomes, and get transcribed, or they could encounter ribosomes that are associated with the rough ER. Now, the mRNAs that are going to actually bind to those are going to be mRNAs that encode for proteins that are going to be either secreted or membrane proteins. The mRNAs that associate with the cytosolic ribosomes are going to be making cytosolic proteins. Okay, so again, just generally understand that the processes are uncoupled in eukaryotic cells. Okay, so the first step that occurs in translation initiation is that the mRNA binds to the small ribosomal subunit. Okay, then the second step is that the initiator or start tRNA has to bind to the start codon, which is AUG, and it's going to be in a specific place to indicate where the protein should actually start. Okay, so now the third step is that the large ribosomal subunit has to bind so that the start or initiator tRNA is positioned in what's called the P site. Over here we have a little ribosome with the sites labeled. So the acceptor site is where the new charged tRNA is going to enter. That's going to be, again, complementary to the codon that's right under the A site. The P site you can think of as being the site where the growing peptide chain is forming. This is about where the peptide bond is actually formed. And then the E site is where the uncharged tRNA leaves. Now, these have different, more complex names in the textbook, and you don't have to worry about that for this class. Okay, so here we are. We've got the ribosome is positioned where the start codon is being recognized, and now initiation is done, and it's going to transition into elongation. Okay, so it's important to remember that elongation of a protein polypeptide is done in the N-amino to C-carboxyl direction. So unlike nucleic acids, which have a 5' and a 3' end, protein polypeptides have an N and a C end. All right, so during elongation, a charged tRNA, I'm not going to draw this, we'll just keep it simple. A charged tRNA will bind into the A site, and it'll be a tRNA that is complementary to the next codon that is in the A site. So GAU would mean that an aspartic acid tRNA would come into here. Then the peptide bond is going to be formed between that tRNA and the amino acid chain that's on the tRNA in the P site. Then the ribosome is going to scoot down and read the next codon. So termination occurs when a stop codon is in the A site. So we imagine that this is the exit here. We would have an uncharged tRNA that would have just donated its amino acid right here. Then we have the tRNA that's in the P position that's got the growing polypeptide chain. And now when a stop codon is in that A position, instead of a tRNA coming in there, there's going to be a protein called release factor that enters the ribosome. So again, this is a protein. And when release factor binds to the mRNA, that's going to cause the whole thing to disassemble. And so there's going to be the free protein, and the mRNA will be released to be able to be used again, as well as the ribosomal subunits. Okay, that should be it for translation.